Okay, so how are you all?
Good, Miss Nandi, and you?
Good. I'm also good. Okay, let's start. Um, so chapter 5, it talks about eukaryotes of microbiology. So basically, eukaryotic organisms who are uh, clinically important, or who are pathogen, human pathogen. We are only interested about those eukaryotic organisms. And this chapter actually is going to talk about those organisms who are clinically important and who are human pathogen. So eukaryotic microbes, every time when we think about, okay, so disease and sickness and all those, we uh, immediately we can think about bacteria and viruses, right? But these eukaryotic organisms, there are a huge number of eukaryotic organisms who are responsible for so many different type of diseases, like malaria. You can say malaria. Malaria, you get malaria from mosquito bites. Okay, so malaria from mosquito bites, but there is one protozoa who is actually responsible for malaria, Plasmodium vivax malaria. So this is a protozoa. Okay, they are responsible for this malaria, and even at this moment 3.4 million people, almost 3.4 million people actually get infected from malaria worldwide. Okay, um, then in US, the number one disease, more than 300,000 American get infected every year. That's called Chagas disease. That Chagas disease, it is actually, if you get a bite from, it's called kissing bug, or it's a triatomine bug, actually, triatoma, or you can say triatomite bugs. If you get bite from this bug, you are going to get, and the disease, it's called American sleeping sickness. You can say Chagas disease or American sleeping sickness. So that disease, it actually affect your digestive, your cardiac, and nervous system, actually. Okay, patient, most of the time patient actually die from the cardiac arrest. So this is the number one disease in America, and the pathogen for this disease is Trypanosoma cruzi. Okay, so if you get that bite from that kissing bug or triatomite bugs, they inject that Trypanosoma cruzi into your body, and you get this disease. Then there is another one, cysticercosis. Cysticercosis, actually, cysticercosis. You get it from tapeworm bite, not bite, that if you ingest tapeworm, it's Tinea solium. Okay. How are you going to get? You can get from your food. If you eat uncooked pork, you are going to get that tinea solium. Okay, so it actually affect your brain, muscle cell. So this is the number two in US. Like how many? I'm telling you the number one and number two based on how many people per year are actually going to die. Then number three, there is toxocara. Okay, uh, it comes from toxoplasma. The pathogen is toxoplasma gondii. Um, it actually comes from the cats, dogs, their feces, their saliva. Um, it actually affects your lymphatic system, then affects nervous system. It's very difficult for the pregnant woman. You know, if there is any pregnant woman and who if that pregnant woman get infected with that toxoplasma gondii, then defective birth occur, defective childbirth occur. At certain point, that child even can die inside the uterus. So all these examples I gave you all are eukaryotic organisms, okay, eukaryotic microbes. In this chapter, we are actually going to talk about all these eukaryotic microbes. Now this organism, this eukaryotic organism, you can understand, like over here I told that tinea solium number two, that's actually tapeworm. Okay, so that's helminths. Okay, multicellular, very large organism you can see with your open eye, versus over here Plasmodium vivax malaria. Or if you talk about Trypanosoma cruzi, yeah, they are protozoa, okay, unicellular organism. So all you can understand that you cannot even compare, you know, like helminths and over here the protozoa, we have algae, we have fungi, lots of eukaryotic organisms are there. There is no similarity. If you go over the morphology, it's a huge it's really diverse actually in morphologically, life cycle, their characteristic features, how they actually their pathogenicity. So everything is really diversified. Okay, so and no comparison. Only similarities between among all these organisms. Just only one similarity is there. That is all these organisms carry eukaryotic cells. Okay, so that's the only similarities among all these organisms. So eukaryotic microbial world, we have three groups actually. We have protist, we have fungi, and we have helminths. Now, under protist again, at this moment, all the organism, eukaryotic organism, we are not done researching, okay, and we have very little information. All those organisms are under protist group. Protist, under this umbrella, under this protist umbrella, there is a huge number of organisms are there, and those organisms, they do not show any relationship with each other. Okay, but why they are there? Because we have very little information about all those organisms. Okay, so we are not done researching yet. So all those organisms we have to classify, we have to keep them somewhere, right? So keep them somewhere. So what scientists did, all those organisms are under protist. Okay, so under protist we have two groups, algae, protozoa. Okay, uh, you can understand that algae, it's a kind of plant. It looks like plant, or you can say plant like eukaryotes. Protozoa, they are always unicellular. So algae, it could be unicellular, very small, or multicellular. Okay, on the other hand, protozoa always unicellular. Okay, so and it's a plant-like organism, algae. Protozoa, they are animal-like organism. Okay, they can move, they are motile, most of the protozoa. So you from this you can see that they are so much diversified, but we really, and the number of these groups, it's so huge. Scientists are, it's not possible actually, it's still we are working, but we have very little information. So they are under protist. So basically protist, they are both unicellular and multicellular. But remember this thing, the protozoa always unicellular. Algae, it could be unicellular, multicellular. Fungi, um, they are again both unicellular and multicellular. So we have a mold. Okay, filamentous structure. You can see those molds in any damp area, and you can see it with your naked eye. So that's the multicellular fungi. Unicellular fungi, um, you frequently use the yeast to bake or to ferment your wine, beer, or your food. So that's the unicellular fungi. Hell means always multicellular and very large organism. They are under this microbial world because most of the time their larvae or eggs, or you can say it's like cysts, they are responsible for our disease. Means these parts only, they are pathogenic. Okay, so that's the reason. And larvae, eggs, they are microscopic. So that's why these helminths, they are under this microbiology. So our first group is protist. Protist, remember I told that you can understand that algae, the plant-like, and the protozoa, animal-like organisms. So they really show very little similarities with each other. So not under a true kingdom, or they really do not have a formal taxonomic term. The organism vary tremendously from one to another, as they do not have shared evolutionary origin. Okay, and that is why this protist is called polyphyletic group. Polyphyletic means they do not have any common ancestor. Okay, basically they do not show any common evolutionary origin. Okay, so no common evolutionary origin. Okay, so that is why they are called polyphyletic. Means they originated from multiple different ancestors. Okay, so that's why polyphyletic. different phylum, or you can say from different types of organisms they have originated. You can understand that algae versus one protozoa. Okay, so that's why the protist, they are called polyphyletic group. They do not have shared evolutionary origin. They are actually not related at all. You know, you can, when I studied my graduate classes, when I took, um, that time, these protozoa, most of the protozoa, they were under animal. Okay, that time, I didn't study about protist. There was no word like protist when I studied. I took my graduate level courses. Okay, so but later, now at this moment, we go by the genetic analysis, right? Most of the time rRNA or DNA sequences we check. Okay, based on that, the scientists, they decide that protozoa, they are not animal at all. Okay, but previously that protozoa, they can move, so that's why, and they don't have cell walls, so altogether scientists used to think that they are animal. But now, by analyzing rRNA and DNA, they are actually protozoa and under protist. So only similarity they have is they have eukaryotic cell, classified according to whether or not the organism is more plant-like or more animal-like. So algae, they are the plant-like protist. Animal-like protist are the protozoans. So when we are talking about plant-like, you can understand that they might have chlorophyll. Okay, they might have cell wall, and so when we are talking about plant-like, then we are going to, they must have cell wall. They might have chlorophyll, not all algae, but most of the algae has um, chlorophyll. Um, and they are non-motile. They cannot move. Okay. Versus when we're talking about animal-like, the protozoans, they are motile. They can move. Okay. They do not have any cell wall. And of course, they cannot do photosynthesis. There are few though, protozoans like diatoms, they can do photosynthesis. Okay, so but they can do photosynthesis. Euglena, they do, um, but the chlorophyll is little different. They really do not have plant chlorophyll like chlorophyll A, B, all those. So that's the difference between algae and protozoans. So um, protozoans, okay, animal-like organisms, because they do not have any cell wall. We do not have human, we do not have cell wall. Okay, we only have plasma membrane, and are heterotrophic nutritions. They do heterotrophic nutrition. Heterotrophic nutritions means organism who derives, okay, so let me write down, who derives their derives their nutritional requirements from, you can say, complex organic substances. Okay, just like us. You can understand that we every time human, we only can consume organic materials. Okay, we cannot absorb, or we cannot take inorganic materials, like you can like carbon dioxide, okay, in air. We need little bit of carbon, okay, for our body. We have nitrogen, high amount of nitrogen in the atmosphere, and we need nitrogen, but we are incapable of absorbing that inorganic complexes or substances directly from the environment. Okay, we cannot. Exactly same way, these protozoans, they cannot. They only can digest or assimilate organic compounds. Okay, so they have heterotrophic nutrition just like us. Okay, and for that, um, because on in environment most of the elements are inorganic elements. Okay, so to get all these organic substances, they need to depend on other organism. Okay, we all the time we consume these organic substances from the plant. What they do, they absorb inorganic carbon dioxide and other inorganic element they can utilize, and we actually get all of those organic compounds from the plant. So that's the heterotrophic nutrition. Now, heterotrophic nutrition, it could be holozoic or saprozoic, and actually saprozoic. Holozoic means it has two parts. One part is ingestion, means internalization of food particles, and another one is digestions, means you can say that processing of food particles or large particles into smaller, and then the assimilation. Okay, then we absorb. So the organism who actually does this type of nutrition, they ingest, and then they inside their body they digest that food particles. That's the holozoic nutrition. Now, saprozoic nutrition, sap means liquid. They actually suck the food. Basically, this group of organisms, that saprozoic nutrition, what happens, they actually consume extracellular, already digested organisms, digested food materials. Okay, so basically they suck or the food particles or nutrients material from outside. Okay, so in this case, they actually get the extracellular, you can say extracellular nutrients material by, you can say, osmosis. Okay, that's the process. They actually absorb, and then they can assimilate inside. So basically, in this case, the nutrients material is already digested material. Means digested means, I'm telling that it's in very simple form. Okay, so that's what this group of organism does. Oh, I forgot to. Oh my God, I cannot move it. Okay, now it's gone. Okay, so that's the saprozoic nutrition. Okay, now this saprozoic nutrition, there is another one, saprophytic nutrition. Don't mix up that saprozoic nutrition and saprophytic nutrition. Saprophytic nutrition, that okay, saprophytic. Okay, in that case, it's fungus, bacteria, they do. Okay, bacteria, fungi, they do this saprophytic. There is a difference between saprozoic and saprophytic. But saprophytic, they actually ingest, or you can say they suck the nutrients material, then inside they digest. They have internal digestion system. Okay, so but over here it is already digested nutrient. So that's the difference between saprozoic and saprophytic nutrition. Okay, so saprozoic nutrition for the protozoans, but saprophytic nutrition it's for the bacteria and fungi. So they are aquatic and terrestrial. You can find them in water and on land, mostly free living. There are a few who are parasitic, like Plasmodium vivax, responsible for malaria. They are parasitic. So microscopic, unicellular, no chlorophyll, no cellular cell wall, all have plasma lemma. Protozoan plasma membrane is called plasma lemma. It's actually the cell membrane. So plasma lemma, basically that's the cell membrane, but immediately beneath the cell membrane, they have another protein layer. It's a protein layer, and that protein layer is called pellicle. Okay, they are responsible for the shape of shape of the protozoa. Okay, and it brings rigidity. Um, the cytoplasm, so if we go inside, they have cytoplasm. Cytoplasm, they have two different layers, ecto and endoplasm. Ectoplasm, it's a gel-like structure. Endoplasm, inside, it's sol-like structure, little liquid. Okay, that gives actually flexibility in shape. Okay, they can move, because you can understand that if they are aquatic, they frequently they can face that osmotic pressure. Okay, so and that, to control that, they don't have cell wall, you can understand. So that's the reason the cytoplasm, they have ecto and endoplasm. Outside, gel layer. Okay, in the outside element directly cannot go in, it controls, and then the sol liquid layer. Protozoa, they are motile, they can move, and based on what type of appendages they have, they are actually classified based on that. They have cilia, flagella, pseudopod, for locomotion. Over here in this picture, you can see the paramecium. They have cilia. Cilia, when it is cilia, it's here like very thin structure and multiple in number. Then amoeba, they have pseudopod. Okay, what they do, they actually, their plasma membrane, they extend. Okay, and they grab the specimen. So let's say over here they have the food. So what ultimately they will do, they will extend this part, they will make a vesicle. Okay, and this extension of plasma membrane is called the pseudopod, and that's how amoeba actually move. So from here they extend over here, and then they pull the body. Okay. Third one, euglena, they have flagella. Flagella, usually most of the organism, if it is flagella for locomotion, one or two. It's a long whip-like structure that actually propel the soul. So these are three locomotory organ for the protozoa. They have cytosome and cytostome and cytoproct. It's like mouth-like structure. It's not actual mouth, but mouth-like. They have an opening over here. Means it's a structure that actually help food to bring in, and this actually anus-like structure. Okay, uh, so this one, this cytostome, it actually do bring food in. Cytoproct, it help to you know get rid of the materials. So it helps to do exocytosis, the process, it's called exocytosis, and this process is called endocytosis.
Miss
And
Nandi,
both,
you said cytoproc. Does that say arms like arms?
no, it's a kind of groove like structure. It's inbuilt structure. It's not organelles, but just like our mouth, they have a groove.
Okay, what did you write above cytoprop?
Cytoproc, it's
Yeah.
like an anus.
Okay. Okay, I see. It was Nandi.
Okay. So these endocytosis and exocytosis together, they are the phagocytosis. Okay. Um, so these two structure, these are built-in structure, but protozoan, almost all the protozoan, they have this structure. Protozoans are heterotrophic. I told you the heterotrophic nutrition, some are holozoic or saprozoic. They have contractile vacuoles. Now, as I told that the cytostome and cytoprop, this is not the organelles, but over here, that contractile vacuoles, it's a membrane bound organelles. Okay, it has you can say membranous organelles. This one, okay. Contractile vacuoles, what it does, it's repeatedly it either contracts or expands. Okay, and by doing that, when it expands, it actually bring in the food or water, bring in substances inside the cell, and when it contracts, it actually expels the internal material to outside. Okay, by doing that, they actually maintain the hydrostatic equilibrium, you can say. So equilibrium. Okay, and that's why this structure actually help them to survive at any pressure inside the water. Okay, so they can tolerate that high pressure, high osmotic, different osmotic concentration, and osmotic pressure. They can tolerate because of the presence of these organelles. And remember, this is the organelles. Okay, so this organelles controls the osmotic regulation of cell.
Miss Nandi, I'm sorry, what was that membrane? Hydrostatic what membrane?
This one, the contractile vacuoles.
Right.
So this is actually organelles.
Okay.
They inside, we have mitochondria, ribosome. So exactly like that, this is a membranous organelles.
Okay.
Okay. So basically this has membrane. And it can expand, expand or contracts. So
okay.
when it expand means it brings in lots of water and minerals. When it expands, contracts, then it expels all the material outside.
Okay.
So based on the water pressure, water level, and based on the the depth or the height of the water.
Okay.
That they can increase or decrease the size of that contractile vacuoles.
Okay.
Okay, so when they are really at the bottom of the water, so it's a huge pressure, right? So that time to protect from that huge pressure, heavy pressure, that these contractile vacuoles, it carries a huge, high amount of water inside, water and minerals. It gives them the weight, okay, and they can tolerate that trash over there.
Awesome.
So that's the contractile vacuoles, and that's how they can control that osmotic regulation, osmotic pressure. Now protozoan, all the protozoan, they have two stage. Okay, they survive in environment in two stage, either in trophozoite stage or in cyst form. These are the two forms they can survive. Okay, there are few a few protozoans are there who actually they can spend their whole life in the trophozoite forms. So few protozoans are there can spend whole life in the form of trophozoite. Okay, but most of the protozoan, so most of the protozoan, they have these two forms. Either they can survive in trophozoite forms or in cyst forms. When it is trophozoite forms, this is the dividing phase, means they can divide, they can multiply, they can grow. Okay, they can, this is the feeding stage. You can say the feeding stage means they can eat. Okay, so basically they can eat, they can grow, and they can multiply when they are in trophozoite phase. On the other hand, when they are at cyst form, they cannot do all these things. They cannot eat, they cannot grow, they cannot divide, or they cannot multiply. Okay, when they are at cyst stage. And these two stages, this trophozoite and cyst, are interchangeable. Okay, interchangeable means the trophozoite stage can convert into cyst stage. Okay, and this is called encystment. Let me write it down. So from trophozoite to cyst, this stage is called encystment, and from cyst, when they convert themselves from cyst to trophozoite, that stage is called, that process is called excystment. Okay, and so basically these two forms are interchangeable. They can change these two forms. Both sexual or asexual reproduction they have. Okay, asexual reproduction, they have schizogony, budding, or binary fission. So these are the three different ways they do asexual reproduction. So the first one is schizogony. So for schizogony, what happens? So if this is one cell, what happened? They do mitosis multiple time. Okay, so from here to here, that's the multiple mitosis. Okay, cell division, but basically the nuclear division. Okay, so they multiply their nucleus, but all the nucleus remains within one plasma membrane. So basically they don't divide the cytoplasm at this moment, only the nucleus division occurs. Okay, and so within one cell you can see multiple nucleus, and this stage or this product is called schizont. So the product of schizogony is called schizont. Okay, now what happened? Then they can live in this form schizont for a long period of time, and then ultimately they do cytokinesis. Okay, when the schizont get mature, so they do cytokinesis, and what they do, that individual nucleus get little bit of cytoplasm and the plasma membrane. Okay, and this stage, then these are called merozoites. Okay, so this is the schizont, means multiple nucleus within one cell. Merozoites, this is the products of schizogony when this schizont get mature after doing after getting the cytokinesis. Okay, so that's the process of schizogony. So you can understand that how from one cell they multiply, they can produce multiple protozoa, okay? And this process is called schizogony or schizogony. The second one is the budding. Budding, what happened for the budding? It's a multiple mitosis, or you can say repeated mitosis takes place at one particular area. So if this is the cell and this area, specific area multiply multiple time, okay? And these are called buds. These buds, they get separated from the parent cell, okay? And individually, they become matured into individual, or you can say adult organisms. So that's the budding. And it happens, budding occurs because of the repeated mitosis at one specific area, or you can say a point of the body. So that's the budding. Binary fission, what happens? So if this is the parent cell, that parent actually divided into two cells, and that's the, so parent first they multiply their chromosomes, and then ultimately they get the cytokinesis, and ultimately parent divide into two. So that's the binary fission. So basically fission occurs, fission. So that's the binary fission. These three are the different three process of asexual reproduction. Sexual reproduction, they have both syngamy, conjugation. Syngamy is just like our sexual reproduction. So what happened? The parent, they produce, so parent produce a haploid gametes. Okay, haploid gametes means N. Okay, and it's a sexual reproduction, so you can understand the two parents are there. Okay, and those two gametes, so N plus N, so maybe this is the female gametes, and maybe this is the male gametes. These two together, they make zygote. Okay, and zygote is the twin diploid. So that's the syngamy. It's exactly like our sexual reproduction. Conjugation, it's little different over here. What happened? So if it is a male protozoa, they are the donor. Okay, and female, they represent as negative one. So this is the female one. They are the receiver. Okay, they receive the chromosome through a conjugation tube. It's called conjugation tube, or sometimes they make bridge, conjugation bridge. So and this is male, okay, and this is female. So that male or positive one, positive means they have a very specific type of chromosome. The presence of specific type of chromosome represent the male one, and absence of that specific type of chromosome represent the female one. So that male, they are the donor, they actually donate the DNA, that specific DNA, specific chromosome, they donate to the female. Okay, and once it donates, then it actually dies. After donation, donating that chromosome, it does not survive, it dies. But the female, it actually, so now N plus N, it becomes 2N, it converts into zygote. It actually acts as zygote. Sometimes it acts as oocyte. Okay, so that's the conjugation. It's a type of sexual reproduction. Now the taxonomy of protists, So till now we were working on the different characteristics of protozoans. Okay, very important. So many questions I can give from those characteristics. Now the classification of protist. So protist, so basically how we classify all these protozoans and the algae. So over here we have, I talked about it that protists, they are the polyphyletic group because they do not have any common ancestor. Over here you can see that all these protists, they are subdivided into 6 supergroups. Okay, we have excavate, chromalveolate, rhizaria, archaeoplastid, amoebozoa, and opisthokonta. These are 6 supergroups. Okay, if you see the origin, you can see that this group excavate, their common ancestor is over here. For this group, their common ancestor is over here. This group over here, over here, over here. And ultimately, all this group, they really do not share any evolutionary characteristics. Okay, so that's why they are polyphyletic group. So, so among these 6 group, we have amoebozoa. Okay, we have chromalveolate, and we have excavate. These 3 groups are protozoa. They only carry protozoa. Okay, and these 3 group are clinically important, means all the pathogenic protozoa are under these 3 group: amoebozoa, chromalveolate, and excavate. Okay, now rhizaria, they have this the foraminiferans and radiolarians. They are also protozoa, but they are non-clinical. Okay, non-clinical protozoa. So we are not going to study about rhizaria because they are not our pathogen. Archaeoplastid, this is the algae group. Okay, opisthokonta, they carry fungi. Few protozoa are there, and mostly the animals. Okay, we are mostly going to study in this chapter, mostly going to work on the excavate, chromalveolate, amoebozoa. We are not going to study the rhizaria and archaeoplastida because algae, there are very few algae are there who are actually pathogenic. Most of them are very helpful. So these 2 group we are not going to study in this course. Or other 4 we are going to work on. So first group, super group, excavate, the Parabasilia. Parabasilia, so under supergroup excavate, we have 3 subgroup. We have Parabasilia, fornicata, and euglenozoa. Excavate, the characteristic features of excavate is the name actually came because there is a depression on the surface of the cell. Okay, because of that depression, we gave them this name, that excavate. So excavate, under excavate supergroup, we have 3 subgroup: Parabasilia, fornicata, and euglenozoa. This group, it has very characteristic features. It might have mitochondria or might not have mitochondria. Okay, there are a few groups who has special type of mitochondria. It called kinetoplastid. Okay, and so sometimes there are few groups who really do not have any mitochondria, but instead of mitochondria they have kinetoplast, and they may or may not have flagella. So based on the presence or absence of mitochondria and flagella, this group actually subdivided into this class, supergroup excavate, subdivided into 3: Parabasilia, fornicata, euglenozoa. Parabasilia, you most of the time you can find them inside the gut of termites and cockroaches. This group, they have just 1 flagellum, They cannot make any cyst. They don't have mitochondria, but they have kinetoplast. Okay, so the kinetoplast, it's a mitochondria-like structure. It really does not do all the work that mitochondria does, but it replaces mitochondria. Okay, this is the energy producing organ, you can say. One example, Trichomonas vaginalis, responsible for trichomoniasis. It is a sexually transmitted disease. It's trichomoniasis, or it's called vaginitis. This disease, so basically, both male and female can get this disease. For the female, for the woman, vaginal discharge, secretions are there. Male, most of the time, for the male, they don't have any symptoms, but they can transfer the disease. Okay, they can carry the disease, they can transfer the disease to someone else. So that's the Trichomonas vaginalis. Fornicator, next subgroup, they form cyst. They don't have mitochondria, but they do have 4 flagella. These characteristics are very, very important. You really need to memorize, okay, all these. What's the difference between parabasilia and fornicator? Parabasilia, they have 1 flagellum. They cannot make cyst, and they don't have mitochondria. But fornicator, they can form cyst, but they do have flagella. 4 flagella. One example, Giardia lamblia. This is the #1 in US, #1 intestinal disease. Okay, cause giardiasis. It's come from the it's basically waterborne disease. Okay, it's a diarrhea kind of stuff. Third subgroup, euglenozoa. Euglenozoa, very important example under euglenozoa is euglena. Okay, euglena, they do have chloroplast. Okay, they can do photosynthesis, but not all group under euglenozoa they can do photosynthesis. Okay, only example is euglena, who actually can do photosynthesis. They have flagella, that's their locomotory organ. So example, Trypanosoma bruzi and Trypanosoma cruzi, they do not have chloroplast. They do have flagella, but they don't have chloroplast. They cannot do photosynthesis. Okay, so I already talked at the beginning, the first slide, I talked about the Chagas disease. Trypanosoma bruzi, that's the African sleeping sickness, okay? And cruzi, that's the American sleeping sickness, or American trypanosomiasis. Trypanosoma bruzi, the responsible for sleeping sickness, you are going to get this protist if you get a bite from CC fly, infected CC fly. Okay, that infected CC fly actually can give you that disease. Over here, sleeplessness, the symptoms, lack of coordination, and ultimately, if you don't treat at the beginning, it's very fatal. Death can happen. Okay. Another one, leishmaniasis. Leishmania, you actually get rashes. It's a skin disease, skin infection. Okay, but if it gets inside the blood, then it's very, very fatal, this disease. Okay. And ultimately it starts from the skin, and ultimately it moves through the circulatory system, if you don't treat it. Next supergroup, okay, so before we go to the chromal violet, I really want to work on the trypanosoma. in this chapter, you have to study few of the life cycle. Okay, so over here we have to study the life cycle of Trypanosoma brucei, that is causative agent for sleeping sickness, African sleeping sickness. Now, this Trypanosoma brucei from tsetse fly, it comes from the tsetse fly bites. Okay, from tsetse fly to human, they actually move in the form of metacyclic trypanomastigotes. Okay, so basically, this infected tsetse fly carry that metacyclic trypomastigote or trypanomastigote. Okay, so when you get that bite, then the tsetse fly injects that metacyclic trypomastigote inside the human body. Then trypomastigote, the metacyclic trypomastigotes actually they multiply by binary fission inside the different types of body fluid. They goes inside the spinal fluid, blood, lymphatic nodes. And ultimately, they convert into trypomastigote. Okay. Now, inside your body, that metacyclic trypomastigote, they actually move through your different types of body fluid in the form of trypomastigote. Now, when a not infected, basically a fresh tsetse fly, when they bites you, they actually get that trypomastigote. Okay, inside the fly's body, the trypomastigote converts into procyclic mastigote inside the gut. Then that procyclic trypomastigote, they leave the mid gut and they convert into metacyclic trypomastigote. Sorry, epimastigote. And that epimastigote inside the salivary gland, they convert into metacyclic trypomastigote. Okay, and that metacyclic trypomastigote is getting injected inside the human body. So these are the life cycle. Make sure you know that what are the stages inside the human body and what are the stages inside the fly's body. Next, the chromal violet. What's the time? Oh, we do have chromal violet. Chromal violet, they have plastid, means they have chloroplast. Okay, they actually carry epicomplexans and the ciliates. Okay, epicomplexan special. They have special structure that's called apicoplast. So basically, this group of organisms, they have their one end of the body has a needle-like structure. Okay, so at just one end of the body, it's very pointed and sharp, so that they can actually get inside the human's body by using this organ. Okay, and this is called apicoplast. So that's the apical complex or apicoplast at one end of the body. And that's their pathogenicity. Okay, non-motile, they cannot move. This is the only protozoa who cannot move. They do glide. They not move. They can do gliding mechanism, but basically they do not have any locomotory organ. Okay, so that's why they are called epicomplexan. Example: Plasmodium. The Plasmodium vivax malaria, you get it from mosquito bites. The non-motile epicomplexan, example, Cryptosporidium. Cryptosporidium, they cause this food. Basically, it's a waterborne disease. Okay, Cryptosporidium parvum. Diarrheal illness, it comes from water. Toxoplasma gondii, I actually talked about it, toxoplasmosis or toxocara. It comes from the cats and dogs poop, you know. And so, and as I told, the defective childbirth, blindness, and they attack the lymphatic nodes. Next group, ciliates. They have cilia, Balantidium coli. It's a foodborne disease, causes dysentery. diarrhea. So you can see that so many different type of protists who are responsible for so many different type of diarrhea. Most of the time the signs and symptoms are similar, okay, but you can understand that pathogenic organism is completely different. Next group, amoebozoa. Amoebozoa, basically previously we used to call them as the amoeba. Two different types. One group they are called slime molds, and another group they are called entamoebas. So entamoebas and the slime mold. So either the slime molds or the entamoeba. Slime molds, look from the molds, the name, it gives you feeling that they looks like fungus. They looks actually like fungus, but for the fungi, they have chitin cell wall. Okay, that's the characteristic features of the fungus. But the slime molds, they don't have that chitin cell wall, and that is why they are not the fungi. Okay, so slime molds, they don't have no chitin cell wall, and that is the reason they are not fungus. Okay, and that's the reason they are under amoebozoa. So that is one group. The another one group is entamoebas, amoeba. So the entamoebas, both actually they are motile by pseudopodia. They form cyst. They have that special stage, the trophozoites. We talked about that already. Example, entamoeba histolytica, responsible for amoebic dysentery, diarrhea, waterborne disease. Negleria fowleri, it's actually brain eating amoeba. I hope you have heard about that, this protozoa, brain eating amoeba. The lake, the pond, swimming pool, little, not warm, but slightly warm water, they resides, okay? These protozoa, they get attracted from the brain secretion, that there is some smell they get, or from brain, some type of chemicals that actually brain secretes. These protozoa, they get attracted from those chemical smell, okay? They enter through your optic nerve, and you are not going to get any smell, and this group of protozoa, they usually eat bacteria. Okay, bacteria is their main food, but when they are inside the brain, inside your body, they actually eat your brain. Okay, so to finish whole brain, it just take few days for them. We really, and brain, you can understand, we cannot inject any medications over there. We can send any medicines inside the brain because of that blood brain barrier. I hope you have already studied that on AP2. So because of that blood brain barrier, it's very difficult to treat any infections in brain. Okay, so at this moment we really do not have. At early stage we still can control, but most of the time the people die actually. So that's the brain eating amoeba. Acanthamoeba, they are responsible for keratitis. So they attack your retina inside your eye, okay, and that gives you blindness. Okay, so now we are going to get into the detail. So amoebozoa, remember I told that we have two groups, that entamoebas and the slime mold. So we already talked about the entamoebas, now the slime molds. Slime molds, they're fungus-like, but they don't have chitin cell wall, so they are not fungus. We have two different types, cellular slime molds and plasmodium slime molds. Okay, cellular slime molds, individual cells remains as amoeboid form. Okay, so basically, in nature, you can find individual amoeboid cell. Okay, then all these amoeboid cells ultimately they aggregate together and they make a slug-like structure. That slug is motile. Okay, they can move, gliding movement they have, and from that slug they can make a bud-like structure. From that bud they make spores. Okay, these are the spores, and these individual spores they convert into amoeba. Okay, so that's the cellular slime molds. Plasmodium slime mold, usually in nature you cannot see this individual amoeba. Every time they actually remains as they exist as a slug-like structure. From that slug, a slug-like structure, they make spores, fruiting bodies, and they make spores over there. They release the spores. From the spores, we get individual amoeba, but immediately they gather together and convert themselves into the slug. So that's the difference between cellular slime mold and plasmodium slime mold. So plasmodium slime mold, every time you can see them in the form of slug or plasmodium. Okay, but cellular slime mold, you can see individual cells. So this is the life cycle of cellular slime mold, Dictyostelium discoideum. Okay, so over here, this cellular slime mold, it has 4 stages. Okay, the first one is the culmination stage, then the vegetative stage, then the aggregation stage, and number 4 is the migration stage. So culmination stage, what happened, that slug or the plasmodium, it makes fruiting bodies. Okay, so basically one sporangium kind of structure they make. Over there inside that sac-like structure or vesicle, they make spores by the process of meiosis. Now that sporangia burst and they release the mature spores and they are haploid. Okay, so from here to here, that's the culmination stage. So basically from slug or plasmodium all the way to the spore production, that's the first stage. Then from the spores they get mature and they actually makes individual amoeba. Now these individual amoeba they actually eat bacteria. Okay, they feeds on bacteria, and bacteria secretes folic acids. And folic acid, that smell of folic acid actually attracts other organisms, other amoeba. Okay, and that's how they aggregate together. Okay, so that's the aggregation stage. So because of the smell of that folic acid from inside of their body of this single individual amoeba, they actually migrates towards each other and they make a slug-like structure. Okay, and that slug they actually migrates at a rate of 2 millimeter per hour. Okay, they just rolled up. Okay, it's almost like gliding. So that's the asexual reproduction of this cellular slime mold. They do, it's not sexual reproduction, but almost like sexual reproduction. Over here, what happened? These two haploid amoeba, so this is haploid, this is haploid. So individual organism, they are actually haploid organism. So they together, they make zygote-like structure, that is 2N, but then this diploid organism or the zygote, they undergoes meiosis and multiple round of mitosis, and it produces the spore, okay? And from that spore, that individual, so they do meiosis, means it becomes, all the spores become haploid, right? So from that spore, individual amoeba actually grows up. So that's the life cycle of Dictyostelium. And this is the plasmodial slime mold. As I told, that every time they remain in the form of plasmodium, okay, or in the slug. So they make fruiting bodies like this. So sporangium or spore. They do meiosis. Meiosis means it's almost obviously haploid. They produce haploid spores. That spores matures and they gives individual amoeba. Now, two amoeba together, they work as a zygote. They are 2N, but what happened? All these 2N zygote, they together, they make a large slug-like structure. That's the plasmodium. This is the feeding body or growth stage, or you can say the feeding stage. Okay. Now this plasmodium in nature, you can see that this is the form you can see. You cannot see this individual amoeba. So that's the plasmodium slime mold. Now the fungi, heterotrophic, saprozoic, okay. Few fungi cause disease, mostly in immunocompromised individuals. Macroscopic fungi, you can see without a microscope, who are multicellular. They are the molds and the mushroom. Microscopic, they are the yeast, they are unicellular. This fungal body is made up with hyphae, long filaments. That's called hyphae. Multicellular fungi's body actually is made up with hyphae. Unicellular fungi also has hyphae, but that's the just one cell long filamentous structure, but unicellular. Multiple of hyphae, okay, so that hair-like structure together make a tangled network, and that's the mycelium. And basically all these hair-like structure together, they make, they are called mycelium, and together they make the body of the fungus, which is called thallus. So there are few fungi who can exist in nature both in unicellular form and in multicellular form. And those fungi are called dimorphic fungi. So dimorphic means they have two morphology. Sometimes in nature, you can see they are actually, they have mycelium, so multicellular structure, but when you inhale the spore, that spore goes inside your body and the lungs, they can exist as a unicellular yeast. They completely unicellular cyst-like structure they make, and they can divide and grow inside your lungs. So that's the dimorphic fungus. Very, very important character that fungi, they have chitin cell wall. The disease caused by the fungus is called mycosis. All the fungal infections are called mycosis. In nature, very few fungus are there who are actually pathogenic. Most of them are very helpful. They are the decomposers. First two chapters, we actually talk about that. We use in our food industry, wine industry, cheese industry. We can get antimicrobial medicines. Almost all the antibiotic, it comes from the fungi. Nowadays we use like especially the yeast we use in biotechnology. Okay, genetic engineering we use yeast. So so this is the hyphae. Okay, now hyphae of a fungi that actually make uh, the thallus of the fungi. Uh, if it is unicellular, like yeast, sometimes what happens because of the budding, you know, budding I already talked about it. So it looks like multicellular hyphae, but actually it's not multicellular hyphae. All the buds remain attached with one hyphae. Okay, in that case, that hyphae is called pseudo hyphae, and we can find it in unicellular yeast. Now for the multicellular, sometimes inside the hyphae we can see the cell wall. So basically this is multicellular, right? So multicellular, but sometimes they don't have cell wall, means they are not separated by the cell wall. Sometimes they are separated by the cell wall. When they are, all the cells are separated by the cell wall, then that's called septate hyphae. When there is no cell wall, but actually they are multicellular, then that hyphae is called non-septate or cynocytic hyphae. We can find inside the molds. Dimorphic fungi example is Histoplasma capsulatum. Okay, they are responsible for histoplasmosis. Very dangerous disease, especially immunocompromised patients, who have AIDS and all those. This is very dangerous. This is very fatal if you have asthma and all those. So this fungi, that Histoplasma capsulatum, in environment, they reside as mycelium, means multicellular fungi. Okay, but if you inhale the spores, then inside your lungs they can reside as a cyst. Okay, and they can survive, they can multiply, they can survive for a long period of time. It's very difficult to kill all those cysts or the spores. So they can survive. So basically in environment on earth, so morphological forms, one is always that can help you to grow, multiply and all those, and the another like cyst and spores, they cannot grow, they cannot have ability to multiply, or they don't reproduce. Okay, so usually the dividing form or the growth form is the main form, main morphology of that organism. That's what we consider. Okay, so this is the only fungus or the only organism, both the main morphologic structure and the cyst, both can multiply, both can grow, both can eat, both are in the feeding stage. Okay, so that's why they are called dimorphic fungi, means they can exist in two different physics or two different bodies, and both is in active form, both can multiply, both can grow, and both can reproduce. Okay, so that's why they are called dimorphic. Reproduction, both sexual and asexual reproduction. Okay, so haploid hyphae, they carry gametes at the tip. So if this is the hyphae, at the tips they carry gametes. Now every time the gametes, they are the haploid. Okay, so when they release the gamete, basically the spores, two gametes, so two different hyphae and two different gametes, these two together, they make the zygote, that's the diploid. Okay, 2n. Now the 2n zygote, it undergoes meiosis. Okay, and it produces then that haploid spores egg, and that's how this sexual reproduction goes. Plasmogamy means the division of cytoplasm, karyogamy division of nucleus. Asexual reproduction either by mitosis, so basically from one cell, just by the process of cell division, mitotic cell division, it can divide into two. We have budding, fragmentation, so basically hyphae, portion of the hyphae, so from here can breaks off, okay, they get separated, and individual piece, so basically from here, this individual piece can grow up and they can convert into adult hyphae. Okay, so that's the fragmentation. Spores, there are so many different types of spores. Okay, conidiospores, basidiospores, so many different types of spores are there. It's in your book. Make sure to study all the different type of spores, the characteristic features. So these are the identifying character of the fungi. So how we actually identify what type of fungal disease we get? We just check the spore. Okay, each fungus has a very specific type of spore. Okay, and that's how we can identify them. So many different, the shape and size actually differs from one fungus to another one. Life cycle, I already talked about it. So they produce spores, haploid spores. So the two haploid hyphae comes together, they make sporangium. Inside the sporangium, they exchange two haploid spores. Okay, from haploid spores, when they mate, they make the zygote. Then from that zygote, actually then it actually does, you know, meiosis. Okay, and from that, after doing that meiosis, it produces that haploid spores. Okay, so basically from the spores you can get two different hyphae over here. Two hyphae, they come together and they exchange the DNA, and ultimately they make the zygote. From the zygote, we get they make a long fruiting bodies. On top they have a large sac-like structure. Over there, that zygote twin, they do meiosis. Okay, they multiply and they make multiple haploid nuclei, and that haploid nuclei converts into spores. So that's the sexual reproduction. Asexual reproduction, we just talked that fragmentation, budding, or just simple mitosis, they can do asexual reproduction. Different disease, rhizopas, on bread you see that black mold, that's the rhizopas. Okay, Saccharomyces, it's very helpful fungus, Saccharomyces cerevisiae. We use to make bread, to ferment beer and wine, in cheese, everywhere we use that Saccharomyces cerevisiae. Candida albicans, one of the pathogenic fungus, causes thrush or vaginal yeast infection, severe skin infection, like for the babies, you know, the diaper rash, that actually candida infection. Pneumocystis carinii, that lung infection, that's you know, mostly for the immunocompromised AIDS patients. They inhale the spores and they actually block the lungs. Dermatophytes, so many different type of fungus together, they are responsible for skin infection. Okay, and they are together called dermatophytes, like microsporum, trichophyton, epidermophyton. They're responsible for athlete's foot, jockey, ringworm. Okay, make sure you know that ringworm, it's a fungal disease. It's not from the worm, not from the parasite. It's not not, it is not a parasitic infection. It is actually a fungal infection. Aflatoxin, actually, this is a toxin produced by the fungus, mostly the Aspergillus flavus. Um, uh, Very, very bad, lots of vegetables, fruits, we can see this toxin. If you see that fungus, it's sometimes it's white, sometimes it's black. It's a really pathogenic toxin that is responsible for liver damage and few cancer. So if you see that you have um, fungus on your vegetables or fruits or inside your food, you really should not eat, because you never know that what type of toxin that fungus actually has created, and they are responsible for cancer. Very, very important for the liver damage. Now the helminths, two major groups, nematode and the platyhelminthes, so roundworms and the flatworms. Multicellular, both has organ. Some are monoecious, means one, aecious means place. So basically both male and female organs are inside one organism. That's the monoecious. Dioecious, both male organism and female organisms, they are separate. Weird dioecious. Okay, but there are platyhelminthes, puas, helminthes who are monoecious because they carry both male and one individual carry both male and female organs. Nematode, Ascaris lumbricoides, intestinal parasites, responsible for ascariosis. That Ascaris, they have sucker inside their mouth. They actually suck all of your digested nutritional materials. Okay, so that person becomes thin, person start losing weight, feeling sick, no energy, and ultimately the person dies. That's ascariosis. Pinworm, Enterobius vermicularis, causes sleeplessness and itching around the anus. Okay, what happened at night? Female Enterobius, they lay eggs. So female lays eggs at night. So all the activity they do at night, and that eats that area, anus area. Okay, and responsible for sleeplessness. It's very, very, very uncomfortable disease actually. Trichinella spiralis, trichinosis. If you eat undercooked meat, larvae encysted in muscles, brain, so fever, muscle pain, digestive system problem. Severe infection can cause lack of coordination because they also affect your brain, okay, breathing and heart problems. If you do not treat it, it's fatal. Platyhelminth is the flatworm, flukes, tapeworms, and planaria. These three groups together, they make platyhelminthes. Flukes, they are non-segmented flatworms. They have sucker and attached to the inner walls of the intestine, lungs, and blood vessels of the liver. They actually suck your blood. One example, schistosomiasis. You get anemia, fever, malnutrition, abdominal pain, fluid buildup, sometimes death. Tapeworms, they are segmented. Okay, suckers or hooks at the scolex. Scolex means that's actually the head or mouth region. They have proglottids. Segments of the body contain reproductive segments. So these tapeworms, their body is made up with different segment-like structure. Each segment is called proglottids. Okay, each segment carry both male and female reproductive organ. Okay, so what happens? So if one segment detaches from the parent body, that segment it actually detaches, and this segment get mature into and adult form, because this segment has everything to survive. It carry all the organs to survive, okay, including both male female reproductive organ. So that's very characteristic features of the tapeworms. Planarians, they are not pathogenic, so we are not going to study about them. One example, at the beginning I talked about it, that Tinea solium, we get it from pork, undercooked pork. Okay, so that's the end of this chapter. And okay, so do you have any questions?
I don't have any questions.
Okay, so we are done this chapter. Lots of things are there. You have to study, thoroughly from the book. So I cannot, talks about everything, every detail. So I made it a little bit short, but there are so many informations in your textbook. So this chapter is very easy, but only thing that you have to remember the scientific name of the pathogen and the connecting diseases, the name of the disease, sometimes the symptoms.
Okay.
Okay.
Do we have lab this week?
Yeah, we do have lab. Today we had lab. So yeah.
Oh, I go Tuesday, Thursday. I was just making sure.
Yeah, tomorrow you have to be there.
All right. Well, thank you.
Yeah. I will see you tomorrow then.
All righty.
Okay, bye.
Bye.
Have a good night.